Hey, folks. So now that we've had coverage of eukaryotic cell structure, apologize for how long that took, but some of the things in there are fairly important to know to be applied later when we discuss other things in other chapters. So I've gone through that, and now we want to talk about the eukaryotic groups of organisms that contain microbes and want to kind of do a survey of those. And so I'm going to go ahead and share my screen, and I believe that's the one I want. Yeah. And let's see. Okay, I think that's good. All right, so let's jump into this. The first part of this I've kind of already talked about, all these different things, so we won't belabor that. We're going to go ahead on through and talk this. Let's just quickly look at this here. So this is a nice table to look at that compares the different kinds of microbes here, the three major groups of microbes that or organisms that contain microbes that are studied by microbiologists. So you can see these major characteristics that could be used to categorize organisms. And so the prokaryotes, you can see the pluses indicate that they have those. In some cases, you see a plus minus that indicates that they may or may not have them. And then you see a few things are qualified with asterisks here, ribosomes, flagella, and cell walls. And in the case of all three of these things in the prokaryotes, the asterisks signify that those are unique to the prokaryotes. The ribosomes that prokaryotes have are special. The size that they have is unique to them. The flagellum is special because it consists of flagellin protein, and it's capable of rotating completely through 360 degrees. You know, there's probably some other things that are special about them, but no other group of organisms have that. And then likewise, the cell wall of prokaryotes, especially the bacteria, is unique. It contains peptidoglycan, and that's not something you're going to find in any other group of organisms. And then size here, with regard to the prokaryotes, is very small relative to other living organisms, other cellular beings, ranging from about a half a micrometer on up to, I think 3 micrometers is a little bit low. Maybe 10 micrometers is more reasonable as a high end there, but the average is still pretty small. And then eukaryotic cells, you can see, range much larger. And with regard to all of these things over here, pretty much in nearly every case, there's a plus, or, you know, it could be plus minus. There's some variation here among eukaryotes with regard to these things like pigments and chloroplasts and so forth. The nucleoid, of course, is missing from eukaryotic cells because they have a true nucleus. So we would expect a negative there for sure. And then the viruses, relative to both of these groups, are incredibly depauperate. They don't have any of these things. And that makes sense because they're not cellular organisms, but rather have a completely different structure. So if we're being strictly rigid in our application of, you know, which organisms are truly living, the viruses don't fall under that category. But because they have some features in common with living things, and because they so heavily influence the lives of living things, they are biological. And so we call them biological particles, okay, if that makes sense. So anyway, you can see the way that these things compare in relation to one another with regard to these important cellular features. And so now we're going to do a survey of the eukaryotic microbes, and these are our major groups. Remember the fungi, that's a kingdom unto itself. These are all members of the domain Eukarya, remember that. Algae and protozoa are members of the kingdom Protista, and parasitic worms are members of the kingdom Animalia. Now, in this presentation, there's nothing relating to the mites, some arthropods, but they too are eukaryotic microbes. But in general, you'll find lots of information on those and other courses. And in the grand scheme of things, their importance is kind of limited. So we're going to stick with these guys here. Okay. And so the kingdom fungi says here 100,000 species. That's likely a gross underestimate. There are many more species than that, but of those that have been discovered and fully described, there's around 100,000. And there are two general groups, those forms that are macroscopic throughout a big chunk of the life cycle of a given species of macroscopic type of fungus. You'll see structures that can be used to identify them, and you won't have to have a microscope. That's not to say that there aren't stages in the life cycle that are microscopic that can be important. There are some, but the large visible structures that they produce are persistent enough and common enough that we'll call them macroscopic. And then we have other forms, again, which produce macro kinds of structures, but are more commonly microscopic. Throughout most of their life cycle, you'll have stages that are microscopic and concealed and difficult to see. So identification of these requires microscopic examination much more frequently than in the macroscopic forms. And these would be your molds and your yeasts, okay? And we'll talk about the differences between these. Up here, these forms, mushrooms, puffballs, gill fungi, all of these different, there's bunches of other common names given these macroscopic forms. What those are typically fruiting bodies. So there's a stage in the life cycle of many of these macroscopic forms where they go from places of concealment, say down in the soil or in the carcass of a dead tree or something like that, what's left over of it. And then periodically, when conditions are right, they'll send out these structures that are visible. These are called fruiting bodies. And so macroscopic fungi are typified by having those. The microscopic fungi typically don't do anything like that. And with the yeasts, they remain fairly concealed most of the time, except when, say, for example, they become really excessive in say, in cases where people have acquired yeast infections and so on, we can see evidence of their presence. The majority of the microscopic forms are unicellular, okay? Especially the yeasts. If they produce larger structures that have multiple cells in them, they're not, strictly speaking, truly multicellular. The cells retain some independence of each other, and so are more or less just kind of massed together without, sharing lots of information and that sort of thing between them. So they're more colonial. And being colonial means that if you were to take a little chunk out of a mass of cells of a particular species and put it elsewhere and provide it with the right conditions, it can grow independently of the source of where the cells came from in the first place. So it's kind of like what we see in things like sponges and other kinds of organisms, where or starfish, you can chunk them up into smaller pieces and they will be able to persist. So they're more colonial. I guess starfish are not strictly, they're multicellular, but you know what I'm saying. So colonial. And relatively little cell specialization. Like in a human body, you have in excess of 200 different morphologically distinct and functionally distinct cells. In fungi, typically there are only at most a couple or three cell types that are, maybe even just one cell type is common for most of them. So they don't have to have specialized cells for different jobs. They don't have cells just for feeding and cells just for reproduction and so forth. So with regard to the microscopic forms, the forms that are throughout most of their life cycles microscopic and need to be viewed with some sort of optical aid, there are a couple of morphologies. So the yeasts, if something qualifies as a yeast, it's very distinctive. The cells are typically round or subspherical, not perfectly round. They might be slightly oblong. And they usually exist independently of one another. They can undergo a process known as budding. That's what's shown here. You can see one large cell. What's happened is it's replicated its genetic material. Some of that's been passed over into this little extension that's forming. And this is a new yeast cell that eventually may break away from the parent cell, okay? Sometimes they'll remain attached for some period of time. And if this cell and this cell and maybe one that's produced by this one, a third cell, remain attached, it'll appear as if they're starting to form a chain. That's not common. But in most yeasts, these remain sort of separate from one another. Another thing about these, this is in keeping with what I've said earlier about a lack of cellular specialization. All of the essential kinds of activities for persistence of yeasts can be carried out by cells that look exactly like this. There are none that are specialized for reproduction or other kinds of functions. And so when they reproduce, it is more or less strictly by budding like this, and that's a form of asexual reproduction. So that's a very simple way to do things. It doesn't require a lot of effort. You don't have to find mates. You don't have to go through courtship and all of that. They simply just divide and their numbers multiply that way. The downside of that is that there's not as much genetic variation as would be the case otherwise. And if you got genetically different organisms that are sexually reproducing and bringing their genes together, you can form new combinations, and that can be helpful when the environment changes and you need to adapt to that. So there's pros and cons to different forms of reproduction. And the pro with asexual reproduction is you can fill a space fast and reproduce efficiently. The con is that you can't adapt so easily to changing conditions. They can do it, but it's not as likely to happen as if they were as would if there was sexual reproduction. Now, other kinds of microscopic fungi, the molds, instead of having cells that are sort of roundish like this and independent of each other, the cells here are kind of rectangular in outline, typically longer. And when they divide, the daughters that are produced remain attached. And that can continue on for some time. So that what winds up happening is they form these long, skinny filaments. And these are called hyphae. The chains of cells that sometimes are seen in yeasts are called pseudo-hyphae. So they're not, pseudo means false. The true hyphae are found only in those forms that have this sort of filamentous look under the microscope, and those would be the molds. And all of the macroscopic fungi likewise have hyphal structure like this. So there are a few kinds of fungi, which interestingly are pathogenic molds typically, that do switch back and forth between these. So there's, if you were wondering, are there any that ever sort of occupy this sort of gray area between those two types of morphologies? The answer is yes, there's a few species that seem to kind of alternate between being yeast-like and hyphal-like this. And these are commonly considered to be pathogenic, disease-causing molds, appear to do that sort of shape-shifting, if you will, between those two forms. But most kinds of fungi are either one or the other, okay? And so those forms that kind of, can kind of sort of oscillate back and forth between the two types of morphologies are said to be dimorphic, which means two-formed. There's two forms within each species. That's not common. Okay? That's not a common thing. Now, one of the things that with all groups of organisms I like to do is think about how they get along, how they make a living. And a big part of that is how they get their nutrition. In other words, how do they get the molecules that contain lots of energy that can be broken down to release that energy to do work, and they can be broken down to release materials that can be used to build new parts of cells. With regard to the fungi, they are all uniformly heterotrophic. So they cannot synthesize their own food at all like a plant or an algal organism would. They don't have chloroplasts. They don't have pigments that enable them to absorb light energy for the purpose of forming carbohydrates. So they must get their nutrition from outside sources. They break down other organisms' bodies in order to obtain that nutrition. And so they're described as being heterotrophic. Now, they do this a little differently than what, say, animals would. Humans and other animals literally have to ingest chunks of food, and they pull it into some structure where then, as it passes through, say, a gut, enzymes are released that help to then break it down. The fungi don't do that. They instead release the enzymes directly. They have at the tips of their hyphae cells that will produce a large amount of enzymes, and those enzymes then are released there at that growing tip, and they literally kind of oxidize their way through the say dead tree, or could be a dead animal, or it could be detritus that's accumulated on a lawn or in soil or something like that. So dead leaves, branches and limbs, and just about anything that was once living, if it enters the environment, when that organism dies, that material, what we call detritus. Let me see if my pen will work. Over here, let me go ahead and share my whiteboard so I don't have to deal with those annoying lines. Detritus. This is the name for dead plant and animal matter. That accumulates in the environment. And there's a lot of that out there. And if it wasn't for organisms like fungi that are capable of producing the enzymes to break this stuff down, the energy that's contained in the molecules making up all of this detritus, that energy would be locked away forever. And the things like carbon and nitrogen and other atoms out of which these molecules are made, they'd be locked away forever in that dead plant and animal matter. But as it stands, fungi, bacteria, and other organisms break it down and re-release those things so that they can continue to cycle in the environment. So that's really awesome that that's how it is. So going back to this figure here, you can see some oranges, and you've got some mold. This is actually penicillin mold that's growing this sort of blue-gray look. and these oranges are slowly decomposing. And this, if this was out in nature, the breakdown products of their decay would enter the soil, and other healthy organisms could obtain, plants could obtain the nutrients being released from that process. So that's different than what animals do. Animals eat mostly chunks of, or whole pieces of organisms. Fungi release enzymes into the surroundings that literally kind of break the detritus down in a remote fashion. And so organisms that do that are said to be absorptive heterotrophs, or saprobes. So let me write that down. They are saprobes. They perform absorptive heterotrophy. And it's like they're slobbering all of their enzymes out into the surroundings, breaking the material down, and then they absorb all of the breakdown products of digestion after that. Other organisms that are ingestive, like us, would take whole chunks of food in and digest that internally, right? So it's a different approach to doing things. Okay. So I don't know how I can stop sharing one thing and ultimately cause other things to stop being shared. I haven't figured that out yet. But anyway, so the organisms that do this are pretty much, that's the main theme among fungi. That's how most of them do things. And that's overall helpful. That's not really harmful to living things when they do this. But there are some few that will occasionally switch over from that mode of nutrition to trying to ingest components of living tissue. So they don't seek out dead things, but they will seek out living tissues and begin then to break those down, do damage to them. So the organisms that do this are not obligate in that. In other words, they don't necessarily always have to do it. They only do it when it's opportunistic for them to do that, right? Mostly they're going to be feeding on dead stuff, but if they get an opportunity to invade the body of a living organism and begin then to break it down, they'll do it. So here then is a case of athlete's foot. The species of fungus that's causing this is called Tinea cruris, Tinea. And you can see there's like a little front here, and it's actually spreading back this way. This whole inflamed area is infected with the fungus, which you can see under the microscope is hyphal. So this is a hypha. You can see the individual cells in it, and they're separated by these little walls. Those are called septa, by the way. And a lot of hyphal fungi exhibit that septal look. Some of them don't have the septa, or if they do, they're partial. So there's variation among species that way. But that's what Tinea cruris looks like under the microscope. And then there are other, this is relatively, it's not without, its complications. And sometimes there can be really pretty severe infections like this. This isn't as severe and potentially destructive as some other fungal infections. There are some that are quite serious. There's a type of fungus called histoplasma. There's several species in that genus, but those can infect the lungs and the respiratory tract, and they can do considerable damage there and even lead ultimately to death. And so some fungal infections are life-threatening, and there are antifungal drugs that can be administered to deal with those. The unfortunate thing about antifungal drugs is that they attack many of the same cell features that we have in our own cells. So it might attack mitochondria or some other activities in the fungal cells, because fungi and humans are both eukaryotic and share similar cell structures. Those drugs that are functional against fungi can also affect us. So even the treatments can be a challenge. Even the treatments can be sometimes quite dangerous when we're dealing with fungal infections. A fungal infection, by the way, is referred to as a mycosis. We got this root word myco in here. We also see that in the word for the study of fungi. There's a word mycology, mycology. That's the study of fungi. So you see the root myco in a lot of words relating to fungi. All right, so here's more on yeasts, and you can see the process of budding taking place here. So the genetic information that's in this parent cell is pretty much the same that's in this bud. In that nucleus, more DNA will be produced and it'll get bigger and bigger, but it's basically the same genes are present there as there are in this parent cell. And this will get bigger and bigger and eventually break away from the parent cell. And that's already happened once with this cell. They can reproduce asexually multiple times. Here's what's left of the area where it was conjoined with its daughter cell at one point in the past. So another cell was produced and broke away here. So that's budding, a sort of asymmetrical division that occurs between these cells. All right, and here's a pseudohypha forming. Like I said, that's not as common as seeing these cells maybe in pairs or as singles, but must be reproducing really rapidly, because as they are produced, they don't have time to break away from one another. So that's a string of cells with basically no ability to share materials between each of them. It's not like things are circulating through from one to the next. They are effectively functionally independent, if not, spatially independent. And then here are some hyphal fungi. You can see these that are forming as the cells divide and this thing grows bigger. The walls between the cells are more or less complete, and so those are called septa. You can see microscopically the septa may be fairly complex in structure. They can have some pores running through them. So information can be shared, nutrition can be shared, water can be shared as it passes through this series of cells from one to the next. Here then is a non-septate type of hypha. And I believe these are much less common than septate forms. But here you have the hypha with no breaks between where cells would be. So it's kind of like each long filament is really a massive single cell that's been fused with multiple nuclei in it. So here it says that penicillium, so this mold that was growing back here on these oranges, that's penicillium. That's what it looks like under the microscope. These are the ends of the hyphae where there's some little cells being produced. These are called conidia. These are used for reproduction. But in these hyphal sections back here, you'll see septa. So these are septate hypha, as is this one. And then it mentions here Rhizopus is a good example of a genus containing forms that are hyphal. So bread mold, black bread mold, is in the genus Rhizopus, and its hyphae would look like this. So that can be helpful in identifying different species. Now, hyphae, for the most part, are going to be vegetative. They will grow as more cells are added to them. Near these tips, the cells actually are especially good at producing lots of digestive enzymes, although other parts of this may as well produce those. And they'll be able to break down, chemically begin to digest a detritus that they're coursing through. Periodically, hyphae from two different organisms may actually meet. So two members of the same species, if they're genetically different enough and their hyphae meet, they can undergo reproduction in many instances. And so those would be reproductive hyphae, where spores then would be produced. Spores are simply single cells that get released. They're very tiny. They can be ejected from the fungus and will typically travel on wind, although there are some species that probably use water for this purpose or some other means to distribute spores. And once the spores settle out in a new place, they can begin to undergo development and form a whole new fungal organism. So there can be vegetative and reproductive hyphae, but typically the differences between those are kind of minimal. So again, there's not a whole heck of a lot of specialization. And so when fungi reproduce, they can do this asexually. And many species do that exclusively, but then a very large number of species will alternate that between asexuality and sexual reproduction. So they can do many of them both. So here we've got a mold. Everything is actually scaled up to make it bigger so that you can see it. In reality, you wouldn't see things quite this large. But this shows a strawberry that's undergoing decomposition. And you see all of the life stages here carried out. And this is asexual reproduction because it begins with a single spore here, which then germinates. So now a hypha is beginning to form as the cells undergo division. And this is mitosis, by the way, the type of cell division that when the cell divides, the daughter cells have exactly the same amount of genetic information as the parental cell had. So that cell divides and becomes this multicellular hypha, which then branches. And this collection of branches, this body of the fungus, if you will, that's made of many hyphae, is referred to as a mycelium. And you can see that mycelium extending here and growing throughout this part of the strawberry that's undergoing decomposition. The fungus is actually hastening the extent of that decomposition as cells are broken down and nutrition is released. And they're taking up that nutrition. And that's fueling even more development of other structures. And here we have hyphae that make their way above the surface. Now, these are specialized for reproduction. So these would be feeding hyphae. These are what ultimately will become reproductive hyphae. And you can see at their tips, as cell division proceeds, those cells start to differentiate, and some of them help to form a covering, while others here form spores that are contained within that little sac. The sac is called a sporangium. And at some point, as it matures, it dries out, it'll open up, and the spores, technically sporangiospores, because they were produced in a sporangium, the spores, which contain exactly the same amount of genetic information as all of the cells in each of these stages here, there's been no change in the amount of genetic information that they have. Those spores can settle out on this particular strawberry, or maybe be ejected and go somewhere else and begin then to break down a new organism. And that cycle will continue. Okay. So by the way, and again, the spore, when it starts developing into all these hyphae, we call that germination when it gets started. All right. These sporangiospores, by the way, these are the conidia that I mentioned earlier in showing you the penicillium mold. In this particular mold, there's a sac around the conidia. In other species, the conidia are basically just attached to one another, growing at the end of this hypha without a sac surrounding them. So those are called conidia. All right, so here's a sporangium typical sporangium, and the sporangiospores contained within. This is also regarded as a sporangium. You can see at the end of this hypha, it's expanded into this little tip. And growing out from it are chains of spores that can be released. But because this is all, one enlarged end of the hypha, it's, technically they're both examples of sporangia. Here we have cells at the ends of these hyphal tips that are not, expanded into a sac-like structure or a big bulbous structure like this. And so the cells that are being produced here are conidia. They weren't contained in some sac initially or attached to some sort of big, massive, fleshy tip at the end of the hypha. So different species will have different ways of structuring these, and you can see a wide variety of things. This could be helpful in identifying the mold species, is to look at these structures, and they're all asexual. Okay. Now, there are many species that undergo sexual reproduction, and they'll produce structures sometimes that aid in the distribution of sexually produced spores, for example. And those can be helpful for purposes of identification and classification as well. And it turns out that among fungi, there's several different phyla within this kingdom. Three of the main ones are identified on the basis of the type of spores they produce. So there's one group called the zygomycota, and they produce these zygospores. There's another group called the ascomycota, and they produce ascospores. And then finally, basidiomycota, another group of fungi, and they produce what are called basidiospores. So we're going to look at the differences between these so that we can be able to utilize that to differentiate the zygospore, ascospore, and basidiospore producing fungi. So sexual spores are helpful in differentiating these. Like I said, there are some species of molds that don't produce sexual spores. They're strictly asexual, some yeasts as well. But when they are sexual, we can identify them on the basis of these structures. So the zygospore producing fungi are in a group known as the phylum, or some people consider it a class, Zygomycota. Okay, let me see if I can write that without creating a big mess on my PowerPoint here. There we go. It's very fickle. Zygomycota. That's the taxonomic name for the organisms in this group. I'm sorry for that little line that shot out across there. But they are so called because they produce these structures known as zygospores. All right. And so let's look at how that happens. Here's a piece of bread, and it's been contaminated, I guess you could say, infiltrated by a fungus. There were some mold spores that were present at the time of slicing of this bread or at some point in its packaging. The spores which were present in the environment settled out on it. And over time, they were able to acquire nutrition and water and so forth, and begin then to form these hyphae. So you can see some hyphae forming from them, and they extend their way through that bread, breaking it down. And eventually, if you have other spores there and they're genetically different enough, they can meet up. So instead of sexes in the fungi, we talk about mating strains. So here's two different mating strains. And what's happening here at the end, you can see where the hyphae are now becoming specialized. So here's an example of cellular specialization. The tips there that meet up will enlarge, and within these you'll have new cells being produced, okay, that are going to be used more or less kind of like sperm and eggs, although there's no difference in size or genetic content. And you'll have fusion of these cells together to form a single cell right here, that within there are actually two separate cells from both of these types. Okay, and so this is an example of fusion of two cells, but the nuclei don't come together. All right. Okay. And so that's called plasmogamy. So if we could look in here and see what's going on in that structure, that zygospore, there's actually two separate little cells in there, and they don't fuse together. They remain separate. All right. Now, as that matures, they will come together in this zygospore. All right. And up from that will grow this stalk, at the tip of which you'll have cells that will be encased in a sporangium. When that breaks open, spores will be released. They will settle out on the bread and begin then to divide and produce new hyphae. But now then, the genetic information in those cells is going to be a mixture of the genetic information that came from the two the two mating types that joined here. So you're taking genetically different organisms and bringing together genetic information from both of them, recombining those, and now then you get a new mating type. You get a new type of fungus as a result of that. So that's what makes this a little better than just strictly asexual reproduction. Now then, the new forms that are produced that are mixtures of genes from previous generations may have traits that make them superior to other organisms in being able to deal with environmental changes that come along later. Okay, so now the fruiting bodies that are formed are typically never massive in these zygospore fungi. These are way exaggerated in terms of their size. What you'll typically see on the slice of bread will be these little tiny black nodules there that you'd have to bust out a microscope or at least a magnifying glass to be able to see exactly what they look like. But anyway, that kind of overviews some of the features of zygospore fungi. And black bread mold in the genus Rhizopus is one of these. Rhizopus. There's several species. That's a common genus of bread mold. Now that group is fairly large, but then a much larger group of fungi, which contains tens of thousands of species, is the group we know as the Ascomycota. And again, it can be considered a phylum by some, others would call it a class. I think those terms, kingdom, phylum, class, those are starting to kind of lose their value as the field of classification advances. The meanings of those terms become less and less evident. But anyway, the particular group that we're talking about here is the Ascomycota, so called because they produce structures called asci, within which ascospores develop. Okay, so these are more likely to be found out in the environment, in soil and other places such as that, in the remains of a dead tree or something. And so here we have one spore, okay, within which there's a single cell that has a single nucleus in it. And the genes in this result in the formation of what's called a plus type mating strain. There's liable to be many different such mating streams. It just so happens they designate this one plus. And it's different from the one over here. This spore gives rise to cells that contain nuclei whose DNA has genes that indicate that it's a negative type. So there could be many other designations. There might be plus plus or plus minus or minus minus or, a whole bunch of different possibilities, not just, two like in humans where we got male and female. The only stipulation for sexual reproduction is that when these hyphae begin to form and they come together, sexual reproduction will only get underway unless they're genetically different. Like if this plus type met up with another plus type, nothing would happen. If this minus type met up with another minus type, nothing would happen. But it just so happens here that they're coming into contact and they're forming a plus type, minus type sort of fusion, all right? And one is going to serve essentially as the female. In other words, this male hyphal tip sends its genetic information over this way, but it remains independent. This is the receiving one. It's got its genetic information and it'll pass it along. You can see growing up out of the female ascogonium is a cell, and it divides and it forms a hyphae. And notice that the genetic material in these cells is separate. Okay, so the two cells have come together, but the genetic material has not fused. So this is an example of plasmogamy here, which I mentioned previously. Now later on, as these hyphae grow, they will at their tips produce these sacs. And within those, you'll have spores forming. And each spore is more or less a mixture of genetic information from these two types here, okay? They're no longer just separate. And what'll happen is these sacs will break open. So this is a whole fungus. It's just showing how the hyphae are positioned in it. Here's what it really looks like. This is a cup type of fungus. It's one of the Ascomycota. There's many other types. You may have heard of morels, morel mushroom, M-O-R-E-L, morels. Morels are in the same group as well as many others. So this is the mushroom. This is the fruiting body composed of all these hyphae. This is basically a mycelium composed of all of these different hyphae. And so these sacs here containing all of these little spores will eventually dry out. They'll burst and they'll release the spores into the surroundings, and they'll settle out, and this whole process will continue on from there. Each sac contains 8 ascospores, right? And the sacs are called asci, and that's unique to this particular group. Notice that back here, when we're looking at the zygospore fungi, there's nothing like that. That sporangium, who knows how many spores it contains? It can have hundreds. Each one is going to contain a variable number of spores. In the Ascomycota, the asci, which are the sporangia, will contain 8 and only 8 nuclei, okay? And they're produced via meiosis. All right. So here you can see in the hyphae that lead to the asci, there's 2 nuclei, and each one of the spores are just a single nucleus. So we've fallen back to that condition. So those are the Ascomycota. This is a big fruiting body. Again, something that you don't see in the black bread molds and others of these zygospore fungi. These are not, massive fruiting bodies, as is the case here. Each one of these is a separate spore-bearing structure contained in that same thing. Most of the life cycle of the fungus will be lived out inside of soil or a rotten log, and they won't exhibit these fruiting bodies. They only do this when conditions are exactly right. Conditions as far as temperature and moisture availability and so forth. And then the third group of fungi, the basidiospore forming fungi, go by the technical name Basidiomycota. Phylum or the class or whatever the expert deems would be an appropriate level of classification, but that's the technical name for the group, commonly called the club fungi. The Ascomycota, the ones we were looking at previously, are the sac fungi. So these are club fungi. Now, you can see that in the soil here, there's hyphae that are growing that arose from spores that were released. So here's hyphae, they grow. Now, if they're genetically different enough, they constitute different mating strains, and they meet up. The cells fuse. They begin then to form this structure that grows above ground. The mycelium forms this fruiting body. You can see it getting bigger and bigger. And it's composed of eventually this stalk. That's called a stipe. Stalk is the stipe. The cap that forms on top, okay, is called the pileus. And you can see underneath the pileus is this series of gills. Some of them have gills like that. That's called a hymenium. Some of them don't have gills, but they have little pores. Looks like styrofoam with little holes all through it. And others are just going to have this smooth sort of surface. More often than not, they're sort of gilled like this. And remember that this is the result of the fusion of cells from hyphae of two different mating strains, right? And here then you're going to have fusion of nuclei and meiosis and all of that kind of stuff going on to produce these little structures here. And see how these little clubs arise along each of the gills, each of these little panels of tissue, and that the tips of those clubs are the spores. These are the basidiospores. And they were produced by meiosis, the type of cell division that mixes and matches the genetic material. And as they are released, as they blow away from that cap, from the hymenium, they settle out in new places, and they then carry forward, the life cycle, start a new generation of hyphae and mycelia. And so the structures that identify these as being club fungi are these little club-shaped things hanging off of the gills, which are basidia. Okay, hence the name Basidiomycota. They look like the tips of Q-tips or something like that, or little clubs, like a caveman club or a wiffle ball bat or something. So those are some of the different variations of fungi. Now, as far as important information for you to know. An additional thing that I like to cover has to do with how these organisms affect other organisms and their surroundings, the ecological interactions, so to speak, they might have. And so fungi are important sometimes for negative reasons, but more frequently are important for very beneficial, positive reasons. So some of the negative reasons, mycoses, when fungal infections occur, those are called mycoses. Things like athlete's foot or histoplasmosis and others. Sometimes people have allergies. So that's not the same as an infection, but this is where you come into contact with some part of the fungus, and due to the fact that its cells have different markers in their surfaces, your immune system responds. And if certain cells like mast cells are triggered, they will produce histamine, which causes your airways to narrow and your blood pressure to drop, and lots of itching and swelling and all kinds of things that are maladaptive. And if that is extreme enough, you can go into what's referred to as anaphylactic shock, which hopefully you will be able to get to a healthcare provider that can administer, say, an EpiPen shot, and that epinephrine will reverse those effects of the allergy. So fungi are different enough chemically from our own cells that their spores and other things are recognized as foreign. And so the immune system goes into overkill in terms of responding to that, and that can be dangerous. That's what an allergy is. Some of them produce toxins, and this may be so that as they penetrate, some detritus, they produce a toxin that's going to keep other fungi and other organisms from coming in there to exploit that same source. So it's a way for them to occupy a territory. That's one possible reason for toxin production. It could be that some of these are destructive, and so it could be used for breaking down tissue so that they can get nutrition from it. There's a variety of things that could be going on, but some very, very deadly toxins are produced by fungi. I'll give you an example. One is called aflatoxin. And it's produced by a type of fungus in the genus Aspergillus. Aspergillus. There's one called Aspergillus flavus, and that's the main one that produces aflatoxin. You can see here's the A. So that's the A in afla, and then FLA is the FLA here. So that's aflatoxin, and it is one that is very very toxic. It can cause extreme toxicity in organisms that ingest it. Commonly, it's found in like soybeans and peanuts and so forth. When the fungus begins to make its way through, say, stored peanuts or soybeans that have been spilled during the harvest process, will sometimes become permeated by the hyphae from this organism. If something comes along and eats those peanuts or soybeans or whatever it is, they may acquire the toxin, and it can cause cell destruction and even death. If you were to be exposed to small amounts of it and survive that, one of the consequences is possible cancer formation. It turns out aflatoxin is also a carcinogen. It's a very, very potent carcinogen, so it can disrupt cell activities to the point where tumors form. So the disease that arises, or the condition that arises, the set of symptoms that come from the exposure to aflatoxin, is called aspergillosis. And of course, there are many, many other toxins that are produced by fungi. And this is why mycology experts always caution people to avoid trying to eat just any old fungus without proper identification. So there's a few things that are hard to mistake for other species. Turns out that there are, among many species of fungi, quite a lot of sort of mimicry, I guess. Different species look very much alike, and yet some are toxic and some are not. So you've got to really watch that. So when crops are harvested and sometimes as a result of poor storage or long-term storage beyond what should be done, sometimes the fungi can make their way into those food stores and destroy them. And so accounts for, molds, for example, account for tremendous amounts of loss of harvested crops like tomatoes and berries and things like that will in short order begin to exhibit destruction by molds and so on if they're stored improperly or for too long. So quite a lot of waste is attributable to fungally infected plants and so forth. Now, the benefits, decomposition, I've already mentioned, recycling of nutrients. There are a lot of different species of fungi that live in soil and are associated with plant roots. So something like 80 to 90% of all plants will acquire fungal symbionts that live in the outer surfaces of their roots. And so these are called mycorrhizal fungi, or simply mycorrhizae. Here's my little thing here. Eraser. I believe there's 2 R's in this. Mycorrhizae. These are like able to grow in, they're hyphae that penetrate the outer surfaces of plant roots and form what some people might commonly refer to as fungus roots. And so here, let me pull up the whiteboard for this. So since we're talking about plants, I use green. All right, so this is the ground surface, and here's a plant. Yeah, horrible drawing, I know. The roots here down in the soil, and they're branching. And if you were to look at a root that has yet to acquire a fungal sort of mutualism, a fungal organism that's going to serve as a mutualist with it, if you look at that root, it just looks plain. There's really nothing on its surface. And so it's smooth and it has a really poor surface area for absorption of nutrition, right? And so the plant, if the entire root system remains that way, basically its ability to absorb nutrients and water is limited to how much branching there is in the small tips of these roots as they grow. But to really drastically improve the performance of the roots, these fungi will enter. And they're not doing that for that purpose. They're entering so that they have the ability to tap into some nutrition from the plant. But as a result of what they do, and I'll show them in a different color. Let me show them as sort of yellow. as a result of what they do, they'll grow and form little tiny extensions, little fuzzy downy extensions out into the surroundings. So the yellow here are fungi that are growing. And if you were to blow that up and look at it up close, you'd see this really thick sort of coating of tremendous numbers of hyphae that spread out into the surroundings. And now if you were to compare the absorptive properties or abilities of that root with the hyphae on it and those that don't have it, the root with the hyphae wins, hands down. It's going to be able to absorb vastly larger amounts of water and nutrition than the uninfected. I'll say they're infected. Usually that connotes a bad thing, but here it's good, uninfected plant root. So like I say, 80 to 90% of plants on Earth's surface do acquire these mycorrhizal associations, and it just makes them grow that much better. If trees don't, for example, acquire mycorrhizae, they may remain stunted. So if, let's say you had a farm where you're growing trees, like a Christmas tree farm, and the soil didn't have the right fungi in there to form mycorrhizal fungi, you're liable to experience low profits because you're never going to produce trees big enough to harvest that people are going to want to put in their homes at Christmas time. So sometimes I think there are ways to inoculate the soil with the proper fungi to develop the mycorrhizal associations, but they're incredibly important. And periodically what will happen as these soil fungi grow out into the soil, they will form these fruiting bodies. And so you'll have you'll have the ability of these guys to produce spores asexually and sexually. So mushrooms that we find in forests are often resulting from the spread of these mycorrhizal fungi. Things like morels, chanterelles, puffballs, other species, all of those are the fruiting bodies of species that are commonly going to be mycorrhizal. So that's very cool. That's very interesting. Okay. So let me see. I have to reshare my screen again. Sorry. So mycorrhizae. There's another name for these that some people use. I can't remember what it is. Ectorhizic or something like that. But I've always just used the term mycorrhizae to refer to those types of fungi that enter the outer surfaces of plant roots and improve their efficiency at absorbing water and nutrients. That's great. Also, we've been able to exploit fungi in numerous ways that are economically incredibly profitable. So antibiotics, we'll talk about those later in the last lecture we're going to do in the term for exam 5. We'll talk about those a little bit. But antibiotic substances like penicillin and chloramphenicol and erythromycin and bacitracin and on and on and on. Most antibiotics are products of the fermentation of other organisms. So fungi and also bacteria produce some antibiotics. Alcohol production, so yeasts are producing things like vodka and tequila and all of that stuff. And even alcohols that are used, topically for, disinfecting skin and so on. Isopropyl and ethyl and other alcohols are products of fungal fermentation. So industrial production of alcohol makes use of these organisms. It's not something that's made in a laboratory. You've got to have living organisms to do that. Organic acids, vinegar, all right, acetic acid and a number of organic acids, some of which are used as preservatives of food and so on. And then vitamins, vitamin production, many vitamins like B12 and others can be produced using fungi. And we can ingest fungi, we can eat them and obtain those same vitamins that way. So they're excellent sources of fiber and other nutritious substances. So yeah, using fungi as foods or using them in making foods, making yogurt and soy sauce, fermented pickles and sauerkraut, all of those make use of not only fungi, but sometimes bacteria, but yeasts are very commonly used in all of those cases. And then, of course, there are some fungi that are outstanding models that can be studied, to learn about how genes work. So one important type of fungus that's been used as a yeast, known as Saccharomyces. Oh, forgot to turn my pen back on. Saccharomyces. Basically the same yeast that is used in baking and so on, that those are yeasts in the genus Saccharomyces. But the thing about them is their cells are pretty large, and the genome, the amount of genetic material they have is smaller. And so when they were working out ways to sequence DNA to learn how genes are put together, they started with Saccharomyces and some other organisms that had smaller genome sizes. So a lot of what we know to be able to sequence DNA in humans and other organisms was learned using Saccharomyces. So that's a type of yeast. Not to be confused with Candida. Candida is the genus that contains the organisms that cause yeast infections. So those are yeasts, but these are infectious forms. So Saccharomyces are the good ones. Candida is the bad one. Yeah, so this says fungi are generally classified according to their, typically it's both of these. It can be sexual and asexual spores. I think the answer that they give is asexual spore. I can't remember which of the two, but yeah, that this quiz question here is actually kind of superfluous, so don't worry about that. It's just type of hypha. It can be all of those things. Type of habitat's really not germane, but these, any of them can be useful in classifying bacteria or fungi. All right, so I'm going to go ahead and stop there before I move on into the protists. And so that'll be another lecture. We'll try to do the protists and the helminths together. All right.